The heckling usually starts later. There was a rather interesting study published in the Great Lakes in Lake Superior, out here in the western end of Lake Superior, and you can see the authors did comprehensive random sampling to look at invertebrate, both planktonic and benthic invertebrate invaders and fish invaders. And what I've plotted here is the cumulative probability of detection as a function of the number of samples required before the species appears in the samples. The first bar here, you can see the line goes straight up. Zebra mussels are world famous for the problems they cause, and they're often very abundant and easy to identify. But another mollusk, it takes this far route, the Asian clam, and in order to have an 80% chance of finding it, you have to have about 80 samples. And if you look at the number of required samples that they calculated for the different groups, if you want to recover 95% of the species in the system, it's simply astronomical. 750 plankton samples, 150 benthic samples, or 100 fish samples. No one samples like that. So that poses a real problem. We also have problems of these false negatives in the lab. And so I had one graduate student, the poor woman, she had to repeatedly count the same sample of plankton from Lake Ontario many times until she became very familiar with it. And then I spiked in 4 different species that are not native to Lake Ontario and which differ in appearance from a very large Daphnia species to a small Eubosmina species. And I spiked them in at different abundances between 1 and 50 individuals. The samples had about 8,000 plankton in them each. And so another thing that she had to do was vary the sampling effort that she used in the lab, whether she was counting a few hundred individuals or up to 8,000, the entire sample. What you can see from these curves is that there's a vast difference between the detectability of Daphnia and Eubosmina in this system. If we compare the red curves, for example, with 25 individuals spiked in, we get a relatively high probability of finding the species with a reasonably low sampling effort. But in the case of the Eubosmina, you have to count essentially half the sample in order to find even one of them, even though there's 25 in the sample. So this poses an enormous problem. We reviewed the recently published literature in the field where people described what their sampling or their counting effort was for plankton samples, and 48% of the papers that have been published over the last 5 years counted 300 or fewer individuals, which would be that vertical yellow bar there, which would mean that in many cases you're never going to see the species that are rare in those samples. So you're going to have lots of type 2 or false negatives. So what should we do? Obviously the title of this conference pertains to barcoding and metabarcoding. We'll go through a couple of examples using aquatic invasive species. There was an interesting case of a species-specific DNA barcoding a few years ago in the Lake Michigan area right around Chicago. Like Kruger, they have silver carp that are on the cusp of entering the lake from the Mississippi drainage. All the fish netting samples suggest that the fish are back here. There's 2 species of Asian carp. This is the silver carp. And there's concern that they're going to swim through this waterway and enter Lake Michigan. A DNA study suggested that the fish are actually either in Lake Michigan or very close to Lake Michigan. And when they published this paper, it set off enormous alarm bells in the United States. The US government argued they provided 4 different reasons why they thought that these results were likely false positives. I think most of those ideas of theirs have been discounted, though. If we look at the use of species-specific markers and eDNA took off really around 2010, and you can see last year, 2016, we had about 33 studies that used this approach. If you look at the actual method that they're using, initially people used conventional PCR, and more recently in red they've gone over to quantitative PCR. Why does it matter? Well, we want markers that are highly sensitive, either markers and or primer pairs. And when we talk about sensitivity, I'm referring to the probability that a species is going to be detected when it is present. And for any of these markers, the relevant thing that we want to know is what is its limit of detection? So I give 3 examples here, marker 1, marker 2, using 2 different primer pairs for 1 and 2. Each has its own limit of detection. And if we look at a theoretical example with a DNA concentration somewhere in the middle here, the concentration is below the threshold for marker 1, so we would get false negatives. But it's above the threshold or limit of detection for marker 2, and so this one should give an accurate and correct assessment that the species is present. Again, we reviewed the literature to see how many people optimize their markers before they go out and conduct their studies, and the results are not that good. If we look at conventional PCR, 48%, and if we look at quantitative PCR, 83% of people do not report or know, perhaps they know, but they don't report the limit of detection in their studies. If we look at whether or not they have optimized the marker, and by optimizing, what we would expect them to do is consider a broad array of markers or a broad array of primer pairs, find the one that is the most sensitive, and utilize that for their studies. And if you look at the results here, it's even more discouraging. 94% of studies do not do that type of homework before they begin their studies. I apologize, this is really washed out. We've been working in Western China for the last few years, and there's a fascinating waterway that was constructed. It's the largest water diversion in the world. It's called the South to North Water Diversion. We happen to be standing on a dock in Danjiangkou Reservoir in 2013, and we noticed that there were some mussels attached to the dock, so we pulled up the mussels, and they were, as it turns out, we were doing a project in South America on the same species, so when I saw it, I knew what it was. It's called the golden mussel. The golden mussel, very hard to see, but it's native to southeastern China and a few other countries in that area. It's not native to Danjiangkou, but as I mentioned, we observed it when we were there. Danjiangkou is the source for the largest of the 3 different waterways that have been constructed. It's more than 1,200 kilometers long, and they're going to shoot water. They built the reservoir super tall, so you would have lots of water pressure, and then in December of 2014, they opened a gate to shoot the water using hydraulic pressure 1,200 kilometers north to Beijing and Tianjin. The problem, of course, is that the species is now going to have a wonderful opportunity to spread north, and so we decided to develop markers and try and track this species as it moves. In this case, we're using CO1, and we developed 13 primer pairs for the CO1. The primer pairs are in blue, and the limits of detection for them are below that. And so the lowest one that we could demonstrate with certainty that we could detect it was primer pair B with a concentration of 10 to the minus 6. And the range of sensitivity for these marker pairs was 10 to the 4. So very, very broad sensitivity, which means that if you chose the wrong marker pair, you'd probably end up with a lot of false negatives. We then did both a series of lab and field experiments. This shows a study where we're looking at detection rate for the 2 markers, B and I, as a function of location. We're going from southern China, where the concentration or abundance of Dreissena zebra mussels, this golden mussel, is very high, to an area in the north where it's very low. And then we have 2 reservoirs in the north that have never been reported to have this species. And unfortunately, this Miyun Reservoir is the destination for the water as it approaches Beijing, and it's a beautiful reservoir, and I suspect it's going to be transformed once this mussel arrives. What you can see here is that both primer pairs were very successful at detecting the species when it was present at high abundance in the water, presumably high DNA concentration. And as the DNA concentration in the water falls, so too does the effectiveness of the 2 markers, until one of them fails, where we start getting no detection whatsoever. So we suspect we are at or below the detection threshold there. And so we were getting some false negatives. And we never had any false positives in our control reservoirs. That work was done using conventional PCR. Here we're comparing conventional to quantitative PCR. This is a location on the east side of Danjiangkou Reservoir, and we have a canal system that's flowing to the east and south, and we were using both of those markers to see how well they could pick up the species. 14 of the 17 sites tested positive with quantitative PCR, only 9 with conventional PCR. The additional sites for quantitative that conventional was not picking up were all downstream where we expect the DNA concentration was very low. And in no case did we have a hit with conventional but not quantitative PCR. A number of years ago, an IUCN group developed a list of the world's 100 worst invaders. And there's a number of aquatic species that are on that list. We've listed them here. They've all been barcoded, but if you look at whether or not the sensitivity of the markers that have been developed has ever been assessed, only 3 out of the 14 species that appear on the list. And these are supposedly the worst aquatic invaders in the world. So we still have a lot of homework to do before we can confidently say that we can use these markers out in the field. I'm going to switch over now to metabarcoding. We had a national network kind of similar to the Center for Invasion Biology here in South Africa. Ours was called the Canadian Aquatic Invasive Species Network. And as part of that network, we were able to conduct expensive sampling using 454 pyrosequencing when it first came out. And we were able to sample across all the port habitats in Canada. I'm not going to address errors that may occur during PCR or during sequencing. What I would like to instead do is focus on problems that may arise during data processing. There's a number of errors that may occur at this stage. The first one, a false positive, where we have multiple sequences of the same species which get put into different OTUs. So essentially, we're inflating the diversity estimate. Second one, a false negative, where the sequence gets filtered out during processing. So we have a false negative, and another false negative where we have 2 species that get lumped into the same OTU. So in that case, we're potentially underestimating richness. So what we did is we took a couple of pipelines, USearch and BlastN, and we took 20 aquatic invasive species at Melania. Christescu had barcoded as part of the Kazan network. And all of the sequences for these 20 species had been tagged, and then we created the mock communities that she described yesterday afternoon. In this case, we populated the mock communities with 50 sequences drawn at random for each species. They were drawn from a pool of between 200 and 46,000 sequences per species. We used 18S as our marker, and we ran a total of 1,050 different combinations of these models, and all models were bootstrapped 100 times. You can see the parameters that we were looking at here. Sequence length with the values that we tested, Fred score, MEE, again, multiple scores, the identity threshold, 97, which is convention, or 98 or 99, or we turned it off. Denoising, multiple states, or we turned it off. And singletons, whether they were retained or discarded. We began with sequences that were 400 to 600 base pairs. Longer sequences resulted in weaker filtering, with weaker filtering, resulted in more sequences kept and higher taxonomic resolution. If we did no clustering or denoising, we ended up with more false positives. I'll talk about that in a minute. If we did either clustering or denoising, we ended up with more false negatives. Our best parameter set of the 1,050 that we tested for detecting an invasive species suggested that we should use relaxed filtering, longer sequences, no singletons, and no clustering or denoising. At the same time that we were doing this, we used the same data set if we wanted to estimate the species richness of these habitats. And in that case, the parameter sets, again, we ran the same number of parameter sets. We ended up with different recommendations, but I'm not going to show that here. In this table, we've shown the top 4 parameter sets for each of the 3 main models. The denoising is D, the no clustering or denoising is NCOD, and clustering only is the C. And the table is organized by this second last column, the number of correct OTUs. Remember, we've spiked in exactly 20 species, sequences from exactly 20 species, and so that's the most that we should be recovering. If we recovered fewer than 20, it was telling us we had false negatives. But in addition to that, we could also keep track of additional species that shouldn't be in there that appeared, which would give us the incorrect OTUs. Now, one can argue, what do you care about most? I would suggest that if you're using metabarcoding to try and do species richnesses in communities, you're most concerned about false positives, because we know the technology tends to overinflate species richness, so I would prefer to err by focusing on false positives. When we're dealing with invasive species detection, we're more concerned about the false negatives. We do not want a species to get in undetected. So we're more focused on this second last column than on the last column. Now, in these graphs, what we've plotted is the number of OTUs on the y-axis versus the parameter set rank on the x-axis. The best parameter sets are to the left side of the graph. The worst parameter sets are to the right side of the graph. And the species that we're trying to find are the blue lines, and the false positives are the orange lines. So if we look, for example, at the no clustering or denoising scenario, about the first 25 parameter sets, we're able to fully recover all 20 species. But you notice that after the first couple, we ended up with very high but variable numbers of false positives. Again, we're not as troubled by false positives because we're doing invasive species work, but still, you don't want any errors. If you look at denoising at first blush, it probably looks better than no clustering or denoising. But if you look at the loss of species, it occurs much more quickly here than it did in the upper panel. And so there's only a handful of parameter sets that we're able to find all 20 species. One of the benefits of denoising is that our false positive rate was very, very low. And finally, if you look at clustering, it's kind of the worst of both worlds, where we lost the number, or the number of parameter sets that were able to recover all 20 species was very few, and almost all of the parameter sets tended to have a lot of false positives. We then used our optimal parameter sets that I just described, and we said, now we want to take real communities of freshwater or marine ports, and we're going to spike in sequences of the same 20 invasive species that I just mentioned. And in this case, we're going to vary the number of sequences that are being spiked in from 1 to 50. And we're going to ask the question, do those sequences make it through the pipeline? And do we get either false negatives or false positives at the end? We iterated this across all of the 20 species. We did them one at a time, and across the 10 ports, and all of the results were again bootstrapped. This heat diagram shows how successful we were at finding the 20 species. If it's yellow, it means we are highly successful. If it's red, it means the models perform very poorly. And even though denoising, in the last round, denoising appeared to be as good or better than no clustering or denoising, when you're looking at real data sets with multiple species and all of which have different abundances, you'll see that the results change quite a bit. By far and away, no clustering or denoising was the optimal route to take, and you'll see that there's similarity between clustering and denoising. Denoising performed the worst, and in some cases, we had a few species in all three of the scenarios where our detection rate was very low. We think we had low quality sequences here. The other thing you can notice, at least for the denoising scenario, I apologize for the tiny letters, but there were some ports where we had consistent problems. All of those ports were on the Great Lakes, so they're freshwater ports on different Great Lakes. None of the marine ports seem to have the same degree of problem. If we look at the number of sequences required to detect a species, here we're doing it by taxon, so we're averaging across the 10 ports. And you can see, looking at the no clustering or denoising, the results are very, very consistent right across the board. We only had one species, a rotifer brachionis, that required more than 5 sequences in order to detect. All of the other ones were down at around 2.9. The other two options, clustering or denoising, perform much worse. You can see there's a lot of variability depending on which species it is that you're looking at. This is the same type of data, but now we're amalgamating across species, and we're looking at each of the ports. And you can see for the no clustering or denoising option, the results are almost invariant as you go across there. And they tick up for the other two, although in some cases clustering performed as well or even slightly better than no clustering or denoising. And as I just mentioned, we required an average of 2.9 sequences to detect an invasive species if it was present using no clustering or denoising. Finally, if we look at detectability as a function of the number of sequences that were spiked in, the solid lines indicate the mean value for the 20 species that we were studying, and the dotted lines, which are hard to see, this is the red dotted line, the green dotted line, no, there's the green dotted line, and the blue dotted line, illustrating once again that no clustering or denoising provided the optimal solutions for trying to find these invaders. We could find 95% of the invaders using by only spiking in 7 sequences per species. If we were using clustering, that value rose to about 11.5, and if we were using denoising, it was up at 43. So no clustering or denoising was by far and away the preferred option for trying to find these early arrivals. In conclusion then, species-specific primers are excellent. I didn't show some data, but we've done other work in China where we've spiked in larvae of Chinese marine species into freshwater samples from the Great Lakes. The larvae had been barcoded. In some cases, we used serially diluted DNA from those same larvae, and the detection rates far exceed what we could ever hope using a microscopy. But even though that sounds like it's very promising, we would argue that you really have to pay attention to the optimization process to make sure that you're putting your best foot forward and you're minimizing the risk of having type 2 errors. Secondly, we can optimize metabarcoding pipelines for different research goals. We argue you should minimize type 1 error if you want to do species richness assessments, and type 2 errors if you want to detect invasive species. If you are estimating species richness, what we found was that slightly more stringent filtering would be suitable. Longer sequences, no singletons, clustering or denoising is okay. And as I just showed, for detecting aquatic invasive species, slightly relaxed filtering, longer sequences, no singletons, and no clustering or denoising. Now we did this work with 454, which obviously is out of date, but we think that it could be adapted for using more modern approaches to metabarcoding. And with that, I'll stop.
Are there any questions?
Thanks for your talk. Is it possible to actually detect false negatives or false positives if you don't know if the DNA is coming from a live or a dead organism? Do you do assessments of DNA transport and eDNA ecology in your studies?
I can't answer that. We would think that the degraded DNA may be more subject to being filtered out, but I'm not a geneticist, so I can't answer that with certainty.
Thank you for your talk. Can you assess the role of endogenous DNA, let's say, in the different ports, and what the ratio of, I'll call it endogenous DNA, the background DNA, has to the detection sensitivity?
We didn't do that, but very clearly some of the ports are going to have a lot more DNA than others, and I think the overriding factor that would affect the results, and it certainly affected the denoising, was that some of those ports would have high species abundances, and they may, because of the way that the algorithms operate, they may end up causing many of the sequences in denoised platform to be thrown out. But We didn't investigate that. We simply went and sampled using the same apparatus, same types of apparatus. We sampled the habitats throughout the country. But if in fact the species that you're inserting are present with only a few sequences, and the maximum we inserted was 50, computationally inserted. We didn't do it to real samples. And they would run a greater risk if there's only a few sequences of being thrown out, if there was in fact a lot of other endogenous material that was already present in those systems. They may overwhelm the pipelines, and so the other stuff gets thrown out as noise.
So, doesn't it, in order to do this accurately, then don't you need to quantify how much DNA is present in total in your raw sample?
Certainly, yeah, I think it'd be nice to know how much there was, just so that you could either account for it or adjust for it if you had to.